Okay, thank you for the introduction, Brittany, and thank you, Remoter, for having me today. Welcome everybody out there to working with sequence data using Biopython in Jupyter. My name's Adriana. I am the instructor for the Python for Genomics course. You can connect with me here on LinkedIn and on Twitter and on my company website. So this webinar is geared towards those who are maybe interested in learning about biological data from a data science perspective. So if you're coming from a programming background, welcome. I will try to explain as much of the biology as I can within this time frame. If you're coming from a science background, just make sure that you have some Python understanding and some data science concepts. And if you don't, I highly suggest that you check out the data science pass, because that's going to give you a lot of the foundation for a lot of this stuff, no matter what you end up specializing in. But before we get to the analysis, I thought that we'd start off by just talking about biological data and genomics in general. Get out of here. All right. So generally, what is biological data and what is genomics? So it's like sequences, DNA sequences, RNA sequences, and protein sequences. So DNA and RNA are normally referred to as nucleic acids. And in this webinar and in my course, I'm going to be referring to DNA. So this is a DNA molecule here. So if you hear me talk about the sequence or the bases, this is what I'm talking about. DNA is essentially the instruction manual for life on this planet, right? There's a story that's contained within the DNA that's explained in bases. So we have adenine and thymine, which have 2 hydrogen bonds, and then guanine and cytosine, which have 3 hydrogen bonds, and they form this helical shape that's, characteristic of DNA. And our job is to kind of understand that molecular story, to translate what that DNA is saying into something that we can understand and to analyze. So how do we get this sequence data? Well, we get it through sequencing. Just remember that DNA can be obtained from any living organism. So in this example here, we're talking about a clinical sample. So we're extracting DNA from liver, but we can extract DNA from any living organism. So bacteria, from mushrooms and fungi, from plants, even from viruses, even though they're not really maybe considered living. But what you do is you extract the DNA or the RNA from the sample. You then prepare it in some way so that you can process it through what's called a sequencer. And there are lots of technologies that actually perform this process for us. That in and of itself is probably worth a whole course, but the output of this process is essentially some computational representation of the DNA sequence down here. So we have this huge file that's semi-human readable, kind of like A, T, C's, and G's, a bunch of letters that are encompassed in this huge file that we then take and put into a data repository. So before we get into the data repository, we have to assess the quality of the sequences. So how well did the sequencing instrument really work to process the nucleic acid? We then line them up or assemble them, because when we take some sort of nucleic acid and process them in the sequencer, we often don't put the whole thing in. We kind of have to chop them up. So we have to assemble them back together, much like you would put a jigsaw puzzle back together. There are pieces that fit. And so we assemble it and then we annotate it, meaning we write down, like where the genes start and stop, maybe some other artifacts contained within the sequence, and then we deposit into the repository. So this is a big data repository that's centered out of the United States. It's GenBank, which is out of the National Center for Biotechnology Information. There are 2 other big ones. So there's one that's in Japan, which is the DataBank, and then the European Nucleotide Archive. And these repositories or databases essentially hold all of the sequences, all of the genetic information that we've collected from all the organisms on the planet Earth that we've researched so far. So as of 2015, we have about 30,000 microbial genomes, about 5K plant and animal genomes, and about 250,000 individual human genomes. So understandably, these databases are quite large, and they keep getting bigger. And the reason they keep getting bigger is because the cost of sequencing has gone down. So I have this graph here, and it looks at the relative cost to sequence a human genome. So if you look back about 20 years, in the beginning of the century, it cost about $100 million to sequence only one human genome. After the first human genome was sequenced, I believe in 2003, the cost has gone down substantially to the point where we're at about $1,000 per human genome, which is pretty amazing, excuse me. The first one to ever get sequenced, like I said, was over hundreds of millions of dollars. So we're at like thousands of dollars for just one human genome sequence, which is, I just think it's astounding. We have the advent of high throughput sequencing technologies. And then we have Moore's law, which is like the computing power, which has been doubling over the years. And that has given us tons of data. But just to give you an idea of how much data I'm really talking about, I found this really cool paper called Big Data, Astronomical or Genomical. And what it does is compare relative areas and their computational demands. Okay, so we have astronomy, we have Twitter, YouTube, and genomics. So astronomy, I mean, by definition, astronomical data should contain a lot of data. To acquire that data is about 25 zettabytes per year. And I have a little cheat sheet down here because I actually had to look this up myself because I'm not as familiar with like large amounts. I'm very familiar with like micro and pico and nano and very small things. But actually, a zetta is 10 to the power of 21. So 25 zettabytes per year for astronomy. To store that data is about 1 exabyte per year. Twitter generates about 15 billion tweets per year, and to store it is about 1 to 17 petabytes per year. YouTube creates about 500 to 900 million hours of video per year and 1 to 2 exabytes a year per storage. Now, coming in at the top, we have genomics with 1 zettabyte per year, requiring about 2 to 40 exabytes per year of storage. And a good frame of reference is like 4 bases is about 1 byte. So that should give you a bit of a frame of reference about how much information we create every year. So we have the grand scale of all this data that we're creating just in just these categories, but just genomics, but we have to think about the future too. So the amount of sequence data or genomic data that we're producing is doubling approximately every 7 months. And I'd like to point out that this paper was written in 2015. So understandably, these amounts have probably gone up. And that's not even taking into consideration the current state of events, which is the pandemic, of course. The pandemic is notable for many reasons. Just talking about data and sequences and within the scientific community, this is the first time that we've really seen data being shared in real time. And what I mean by that is before this pandemic, and in this Washington Post article, they go into a little bit more depth, but they are referring to the SARS pandemic, which occurred in the early 2000s. We're talking about releasing sequences and publications that would take weeks and months. You would have to get it peer reviewed, and you'd have to pass basically all these gates to get things published. Well, now we're opening a lot of these gates so that data is just being released in real time, instantaneously. And I think a good quote from this paper is that it's, imagine walking from Chicago to San Francisco, and then imagine taking a plane to Chicago to San Francisco. That's the kind of difference. So that's the kind of difference that we're talking about in terms of genomic data. From a personal perspective, just watching researchers on Twitter, I'll watch like sequences being released one day, and then the next day these sequences are being deposited in the repositories and then automatically uploaded to these applications that are showing us mutations in real time in phylogenetic trees. And it's just a really amazing time for the scientific community coming together. So that's really what we're talking about. And I hope I've convinced you that genomic data is really one of the bigger players in like the quote unquote big data revolution. Because you have scientists and researchers and data scientists all coming together, trying to collaborate and figure out, what do we do with all this data? How do we store it? How do we analyze it in an efficient manner? And how do we make sense of it all, essentially? So I'm going to narrow down a little bit and talk about genomic data from a data science perspective. And one of the tools that I do actually use when working with sequence data or genomic data. So genomic data is not any different from, like sports data or financial data. From a data science perspective, it does follow the same generalized steps that you would see in, data science curricula, and that you have a hypothesis or some sort of question that you want to answer. You have this idea that you want to explore, and then you acquire the data, which is the first step. You clean the data or pre-process it so that you can then analyze, and then you have to communicate your results in some way. So you would have some sort of visualization or some sort of written summary. I like to use Biopython, which is the core of this webinar for the first two steps, so data acquisition and data cleaning. I will try to take you through all 4 steps in this webinar, although I'm going to have to step out of Biopython a little bit, and I go into more depth in my course, and I'll walk you through an entire process. Okay, so what exactly is Biopython? It's an open source set of tools that make working with biological data more streamlined. Of course, it's written in Python, and it works with the essence of bioinformatics data, which is the sequences and then the related metadata. It really excels at converting files. So when you have lengthy bioinformatic pipelines, you're often hopping over from application to application with sets of data. Often one file format is not compatible with the next application. And so Biopython is really good with those intermediary steps when you need to do the file conversions. It's also really good at parsing large files, so it knows how to separate sequence data into workable pieces for your code. It can perform various basic computations, such as weights of molecules and converting DNA to protein, the minutiae that we really shouldn't be worried about when, developing grand scale applications and scripts. It expedites work with biological data repositories, so GenBank, what I showed you earlier, in NCBI, so if you want to download sequences. It also allows you to download publications and then protein sequences from SwissProt. Biopython also does some specialized tasks such as running alignments, so lining up portions of DNA. And it can also build phylogenetic trees, such as this one, where we're just looking at relationships between organisms and seeing how similar and different they are. Okay, let's jump into the data set that we're going to be exploring, which is the novel coronavirus. And so I'm going to preface this with the fact that I will move through the analysis really quickly just to get you an idea of what working with genomic data is like. And for this project and for the course, we will be working exclusively with viral genomes. They're much smaller, less computationally expensive for us to work in a local environment. If you were to work with human genomes, you're on the scale of 3 billion base pairs, likely you have to work on some sort of server in the cloud. Viruses usually are a few orders of magnitude smaller, so phages are usually a couple thousand bases. The largest virus is only 1 million bases. Okay, let's go over some nomenclature, and we have the novel coronavirus is known as the severe acute respiratory syndrome coronavirus 2. So you'll hear me call it SARS-CoV-2. A lot of people also call it SARS-CoV-2. The subsequent disease is coronavirus disease 2019 or COVID-19. So here is the virus itself with its characteristic spike glycoproteins on the surface of the virus. So that's what's interacting with our cells to facilitate entry and infection. We have other proteins that are contained within the virus, such as the nucleoprotein membrane and envelope. We have a genomic representation down here. So we're looking at the genome right here. And I always really like these pictures because the color always matches. So you can kind of see where the genes are placed within the actual virus. So this blue region here is called Open Reading Frame 1AB. Open Reading Frame just designates an area that hasn't been delineated into specific genes yet. So it's usually like a large expanse of genetic information, and that's enclosed in, it looks like it's in the envelope. So you can kind of see like some blue behind there. And then the S gene is encoding the spike glycoprotein, which you see on the envelope here. And then we have the other genes and areas down here, which are also color coded. So the size of the virus is around 29,000 bases. It is one of the larger viruses, and it's a RNA virus. So for the sake of this webinar, I'll always just say DNA, because typically that's what we work with in NCBI. That's what's deposited there, and that's what's represented as a DNA molecule, but it is in fact an RNA virus. Okay, so let's say that we have some paper here, rapid detection of novel coronavirus or SARS-CoV-2 by reverse transcription loop mediated isothermal amplification. Really long title. This is a publication from which I went ahead and saved some quotes. So the gist of this paper is that these researchers want to develop this diagnostic test. And they ran all these tests, and they basically isolated these regions in the the genome that targets these genes. And by developing this test, they're able to detect the virus in clinical samples by just targeting this gene. So what we're going to do is look at the two genes that they isolated as being conserved and mutated. So what's the difference? So we want a gene to be relatively conserved when we develop some sort of diagnostic test, because we want our test to reliably and reproducibly detect the same region over and over again, no matter what happens to the virus. If we select a region that's mutated, meaning it has a lot of mutations, then our test will inevitably fail. So we're going to go and check out two regions. So we're going to check out the S coding region, which is yellow up here. And that one is known to have a lot of mutations and therefore was avoided in this paper. But we're going to kind of do a contrast, and we're going to look at the non-structural protein 3 region of Open Reading Frame 1AB, which is up here. So we're going to look at the S coding region and the NSP3 of ORF1AB. And we want to answer the question, what is the relative conservation of those areas? Can we compare the rate of conservation with one gene with the other gene to kind of push us in the right direction when we want to develop our own diagnostic test? So to get this number, we need to get all the genomes. We need to kind of clean it all up and perform some calculations to see if we can look to find if there are any areas of conservation. So this actually looks like something that I would make myself. This is much neater than what I would actually do. But basically just piling a lot of publications in Jupyter Notebook, and then links and papers, and then kind of going down with my analysis down below. So I have what's called a ref-seq, or a reference sequence, and that's essentially a point of reference for us, and it's a reference genome for which we can kind of derive a point of reference. I know I just said reference a lot, but what we need is some reference that we can kind of say, okay, well, I was using this reference sequence, so we can all work together and go from this point of reference and do our own research. So I have a reference sequence here, which was given to us in this paper. And let's look at a complete picture of our workflow. So remember, I went through the steps above. So we have acquisition, we have cleaning, we have analysis, and visualization. So these are the 4 steps, and then I've color-coded the area to tell you where we're doing that process. And then the pink is indicating the input and output. Okay, so we need to get accession numbers from a database. And what accession numbers are basically like a barcode for the sequence, so that I can go into any kind of database and retrieve the sequence and its metadata. So I'm going to grab a bunch of accession numbers. Then I'm going to use the Biopython's Entrace package, which talks to an API at NCBI and will download batches of files for me, the sequence files that we need. We're going to put those on our notebook in the form of a GenBank file, and then work with some Biopython scripts to clean and separate the data. So I only want data that has the S gene, or a data set that only has the S gene, and a data set that has the NSP3 gene. And I don't want anything else. I don't want the other genes. I just want to look at the conservation rate between these genes. I'm then going to take that data set on both ends and then run them through an alignment. And if you're not familiar with an alignment, what it does is it uses an algorithm. It's pretty computationally expensive in that it takes all of the sequences and lines them all up so that they all match to the best of what the algorithm can figure out. And what it does is it computes a consensus sequence, or the best representation for all of the sequences that you aligned. So you take your alignment, and then from that you can get a summary, you can derive your consensus, like I said, you can get a conservation, and then from that we're going to use a little bit of pandas and NumPy to generate a conservation plot. All right, so let's start with data acquisition. So I got my initial data set from NCBI virus, virus variation, and I was gonna see if I could show you, but it's essentially a database that, let's see if I can get out of here. Here it is. Okay, yeah, NCBI virus. So if you're familiar with anything that's related to data science, I feel like you're always moving like data from one place to another place and then cleaning it up and then moving it to another place. This is my starting point. So I will extract all the metadata from this database, then take that information and push the accession numbers to another database to extract the information that I want. And so I have a bunch of parameters that I can specify to filter out all of these sequences. So each one of these is a sequence or a clinical sample, or a sequence that's derived from some clinical sample that represents a coronavirus. Like for instance, this one was released in early January, which means it was really early on in the pandemic, from China. It has the length of 29,000 base pairs. It was isolated from Homo sapiens sapiens, which is humans, so on and so forth. So I have this huge database. Let's go back. I went ahead and extracted that file. I filtered by these constraints here. I wanted complete genomes because I want to be able to pick and choose what I want. I isolated specifically from North America. I'm just making this up just for the sake of this webinar. And I isolated sources, clinical samples only from the oronasopharynx, which is behind the nose and the throat. So that's technically where you should be getting your nasal swab from. And then the collection date is mid-April to mid-July. So I find the easiest way to start is on my own terms, which means I want to import everything in a data frame that I can manipulate. So I use pandas and NumPy, read CSV function to read in my file that I downloaded from virus variation. And here is the data frame. So it's basically looks like what we just saw on the website. And then I like to always do a little bit of rudimentary analysis in the beginning to kind of get an idea of what this whole data set holds for me. So I think it's always interesting to look at where all the samples are coming from. So I went ahead and looked at all the unique identifiers for the geographical location column. So we have all of the unique ones here, and then I use Seaborn and Matplotlib to kind of plot it out for me in a semi-beautiful way. Not really, but just something to give me an idea of what I'm looking at. And it looks like we have samples coming from all across the United States, with Wisconsin-Dane County doing their due diligence and depositing their sequences probably exclusively in this database. The thing about sequences and where to submit these sequences, there's never like a defined place. We don't have a defined place. Like I said, there's more than one database, so sometimes there's more than that. We'll deposit it in more than one database. So this graph right here, doesn't represent all of what was deposited during that time. It just so happens this is where these particular hospitals or labs decided to deposit their sequences. Okay, so what we're interested in is the accession number. Remember I said that was a barcode? So we need to get all the barcodes, which is this column here, because I'm going to use that to feed to NCBI to get our sequences. So I just went and I extracted it and saved it to a list, and I have 502 sequences that I need to extract. So to extract, I'm going to use the Entrez package, which is from Biopython. So Entrez is a web API from NCBI. It allows us to interact. You can either use the terminal or you can use Biopython here, but it allows us to go to their databases and efficiently download sequences. So if you think that you have hundreds or thousands or hundreds of thousands of sequences, to manually download, click, 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 all through those sequences, just, it's not going to work. So the API allows us to just, write a few commands, and then it'll batch download the sequences and, plop it right on our doorstep. It is a publicly, NCBI is a publicly available resource, so we have to be courteous to others. We have to be courteous to them. And so there are a few kind of ground rules that everybody follows. The first one being that we always specify our email, because we always want them to contact us in case there's an issue. We don't want to make more than three requests per second, which Biopython actually takes care of for us. And then we don't want to do huge downloads during business hours. So anything that you have really large, try to do in the evening and on the weekends. We're working with viral genomes. On the grand scale of things, that's not anything to be worried about at all. Okay, so import the package from Bio, specify the email, and then I'm going to create a new file. So all the files when working with Notebooks AI and Jupyter Notebooks are going to be over here on the left. So I'm going to create a file in the data folder, and I'm going to download the reference sequence first. So remember how I said the reference sequence is kind of like the, it's the reference. So we're going to investigate just that one sequence and then go in and kind of figure out where all the genes are and the locations so that we can apply that information to our batch data or all of the 502 sequences. So I created a new file. I saved it over here, or it's going to save over there. And then when I do the eFetch, you pass in a couple parameters. So the database, which is NuCore, or the nucleotide database. So NCBI has so many databases. It's not just sequences and things like that. You have sequence read data that comes straight off the sequencer that goes in there. You have publications, you have protein sequences, and then their structures and everything like that. It's insane. So there's actually a lot of functions that are really cool within this package. I'm just going to go over these two, but you could explore this even further. But anyways, the database is NuCore. Then we specify the ID. The retrieval type and mode are optional, but I always like to specify that I want my file downloaded as a GenBank file and in text format. So it returns a handle, which we then read and write to our file and then close everything up. So here's the file right here. You can double click on that and look at it. I usually like to do everything in notebooks so I keep track of what my brain was doing at the time. So you can exit out to the command line, go into terminal using exclamation point, then bring up the file. And this is what a GenBank file looks like. This is exactly what it looks like when it's online too. So you have all of the metadata up here regarding that reference sequence. Here is the ID or the accession number. We have the length, single-stranded RNA. If it was associated with some publication, we have that information here. We have something called features. So remember how I said that we annotate or write down where things, genes start and stop, untranslated regions. So other annotations are all listed in the features attribute within this object. Here we have a protein sequence, which kind of just looks like, doesn't look like anything much. That's what I meant by something being semi-human readable, and that it's characters, but to me that doesn't really mean much, at least in this context. all the way down here, we have more features, and here is the DNA or RNA sequence. So 29,000 bases is quite long. I'm just going to scroll all the way down. And we'll move on to, now that we have our reference sequence saved, we're going to move on to downloading all of those files. Okay, so the way to do this with Entrez, when you have a large amount of files or files that are contain a lot of information in them, you're going to want to split up the download into batches. And so with Entrez, you can use an ePost function here. And what that does is perform a search up on their server, saves the search results, and then we then retrieve some information from that search and then use it in our eFetch when we fetch the sequences from the server. Okay, so let me show you how to do this. The ePost function, we're going to specify the database in which we want to perform the search, which is the nucleotide database, and then the ID, which is all those accession numbers, and the ePost functions knows how to parse this so that it searches for each individual sequence, save it to a handle. Then we use the read function, save the search results. Now from the search results, we want to save two parameters, the query key and the web-env. Those parameters are what specify this search result and is what we're going to use in the eFetch to download all the sequences. So in this loop, we're going to specify a batch size. So you can customize this to the size of your files and the amount of your files. I'm just going to go ahead and do 50 at a time because they're viral genomes. They're not that big. I'm going to save this to a new file in our data folder, and then it should be in the output folder, and then the count is going to be the total number of sequences within that list, and then run a for loop that's automatically going to count for me and then give me a status message down here, and then it's going to eFetch using all the parameters that we kind of specified before. So the database, retrieval type, retrieval mode, two new ones are start, four new ones, sorry, start, the batch size, and then the two parameters, or sorry, the two variables that we saved up here, the web-env and the query key. We will then read that, close everything up, write it, and then close the whole thing up, and then give me a smiley face to tell me that it's all done. So we should have our file here, which is the SARS-CoV-2 oronasopharynx GenBank file right here. And I just went ahead and looked at the top of the file just to make sure that it had all of the, or at least just started to collect all of the sequences that we needed. Okay. Let's go down to the next step. So we have our files, we have our reference file here. Out here we have our reference sequence, and then we have the output, which is here. So we're going to go ahead and do our prepping, cleaning, and sorting this. And so we're going to use Biopython, the seq function, the seq.io function, and this import the seq module, seq.io module, and seq record modules to create new objects. Okay. Let's go ahead first and open up the file that contained all of the sequences. We're going to use the parse function for this one because what it does is give us an iterator instead of opening all these files, because these files can be quite large. We're going to use an iterator and count just to make sure that we have 502 sequences, which we do. Then I went ahead and opened up the reference sequence, and for that, we use the read function because it only contained one record in it. So that's the only time you can really use a read function. And it returns the seq record for me here. And so this is what the reference sequence looks like in Biopython. So let's explore the reference sequence. We want to search for, remember, we want to search for the S gene and the non-structural protein 3 of open reading frame 1AB. So let's do the S gene first. But before we do that, we have to go in and look at the reference sequence and see what kind of genes we have, what kind of features. So the genes are annotated underneath the features. So we're going to go ahead, and I just wrote a small for loop to write all of the features out for me. So these are all seq feature objects. And annoyingly, when you have a seq feature like this, it actually doesn't give the name of the feature or the gene. It just gives me the fact that it's a gene and its position. But I mean, that's not really helpful to us. It's never helpful to me because I don't have these things memorized. So you have to go one step further and go deeper into features. There's an attribute called qualifiers. And if you look under qualifiers, there's a dictionary that has the name gene. And if you print all those out, that actually gives you all the gene names. So these should all look pretty familiar to you now, because this is what we saw on the genome diagram, right? So it's actually all in the same order too. So we have open reading frame, then S gene, then open reading frame 3, envelope, membrane, so on and so forth. Okay, so we want the S gene and we want the open reading frame 1AB, specifically one protein within there. Okay, so let's start with the easy one first. The S gene is we just want that whole yellow part. So let's go ahead and extract that from the GenBank file that we just downloaded. So we want to move through every single record in that entire file, extract out the features from every sequence. So the gene is equal to S because that's the name of it. This is what that feature looks like right here. This is what a seq feature looks like in Biopython. Now, if we want to run through all of the records in that file that we just saved, we just say for every record that's in this COVI-ORO iterator, for every feature that's in this record's feature list, if the feature type is equal to gene and the qualifiers gene key is equal to the S value, create a new record, append that to a list up here, which I have 502, write that to a file. Theoretically, we should have a FASTA file that contains 502 S genes from that entire data set that we got from virus variation. Okay, so now we're going to do the same thing with the non-structural protein 3 gene. We have to go a little bit further on this one because although the open reading frame is considered an artifact or a gene, we have to go one step further inside that feature. And I will show you. We want to, instead of looking for the gene key, we want to look for the product key. And I go way in depth into this in the course, but we basically go into the features objects and kind of explore from there. And you kind of learn the anatomy of all of these objects and kind of learn how to extract information that you need. So I went ahead and explored that further and extracted the product that was equal to non-structural protein 3. I went ahead and just printed what the seq feature looks like. And this open reading frame is actually really interesting because it has 2 copies within the same record. And this can mean a couple things, but I'm not an expert on novel coronavirus, but I do believe it probably has to do with the reading frame being read differently. It could be another reason, so if you're an expert on this stuff, feel free to correct me. Okay, so the non-structural protein 3 is 5,000 bases long. And then we're gonna do the same thing. So we're just gonna move through that file, move through the record, search the features. We're gonna specify the product instead of the gene. And because I only wanna extract the first one in every record, I just went ahead and created a list and then double checked if that record was not already in the list to append it to our actual list up here. So we have 502 sequences that were contained within this list, and then I wrote that to a file. Okay, so now we have 2 files. We have the S gene and we have the NSP3 gene, which are contained within 2 data sets and 2 FASTA files that we can do whatever we want with. So at this point, I usually hop off of Biopython and work with other applications. The one I'm going to talk about today, not really going to go into it in great detail, is ClustalOmega. And ClustalOmega is an alignment software. So like I said, alignments are typically pretty computationally intensive, expensive, because you're lining up multiple sequences, sometimes hundreds, sometimes thousands of sequences. So Biopython has a wrapper for this. So you can do small alignments here within the notebook. You can go on their web server and do your alignment there. You can do it locally. If you download the application, you can run the application through your terminal. Okay, so I went ahead and I did the alignment. So we're lining up all the genes to make sure that they're all lined up. And then I'm extracting the summary and I'm going to import it into here. So to work with these summaries, Biopython also has a package for this called Align.io and Align.info. So we can read in alignment files and extract just the information we need. Alignment files can be rather large because it just, you're aligning all of these sequences, has all this information, this raw data. And so a lot of times we don't really need to work with that entire file, but we just need to work with the summary of that file. And that's what we're doing here. So we read in the Clustal file and we convert it to an alignment file. So Align.io is similar to Seq.io in that it has a lot of similar functions. So we read in files and we can parse files. Now from that alignment, I can calculate a consensus. And a consensus sequence is the most likely base for that position. So it's a representative sequence for an entire alignment. So if I were to choose the base that was most popular for that position, I would assign it down there. And then what you get is this long sequence that represents your alignment. So I can run a method called dumb consensus, and that's just a really quick and dirty way to get a consensus. There are more complicated ways, which is why it's called dumb consensus. And then we're going to hop a little bit into Scikit Bio, which is another bioinformatics package. Now that one, I just think it's a little better at doing more of the computational stuff than Biopython. It's not that much more difficult to learn, but we do have to do some file conversions, and it's readily easily to do if you just use the convert function that's furnished by Biopython to convert the sequence alignments into tabular MSA format, which is what Scikit Bio uses for alignments. Now, an alignment for Scikit Bio looks a little bit something like this. This is obviously a truncated version. If we're aligning 500 sequences, it'd be 500 sequences all lined up. But this is what that object looks like. And from this object, I can do a couple quick calculations. So conservation, remember, we wanted to look at how conserved the sequences are, how similar they are, because that's where we want to design our test. So we can actually run a conservation method. So we just specify some parameters and we get an array where the 1 is equal to 100% conservation, or the base is 100% the same across the board. So anything less than 1 is going to indicate 98% conserved, 70% conserved. You can then calculate the total amount that's conserved over the length of the gene. So basically summing all of the numbers in this array that are equal to 1. And that gives me about 3,400 bases. And when I divide that into the total length of the gene, that gives me the percent conserved over the total gene. So we know that the S gene is about 91% conserved, which relatively speaking is not conserved when we're talking about viruses. Okay, so I did the same exact thing. So I just did everything all the same, except I just fed in the alignment from the NSP3 gene file, or the ORF1AB. I know I call them two different things, but the Open Reading Frame 1AB file, the one that should be more conserved. So same thing, we have to convert it. We look at the conservation, calculate the total amount conserved, and we find that this one is 95% conserved, so more conserved than the S gene, just like the author stated. So this is kind of pointing us in a similar direction. it's not like an end all be all, but it would definitely be something that would be worth investigating if you were planning on designing primers or designing some sort of test to isolate a region. For visualization, I thought I'd show you just something pretty easy. There are obviously more complicated and more beautiful ways of showing this. You could use Plotly to show like dynamic plots and move around, or you can move outside of the notebook, and there's actually specialized software just for this purpose. But I just thought you would know about some capability within notebooks. So we're just going to use Pandas and NumPy. And it's just easy because we have some arrays up here, so we could just feed the arrays into a simple graph. So I want to show the amount of conservation across the gene. And to do that, I'm just going to import the consensus. So the most likely base for that position into a data frame. And then I'm going to import the conservation score across the length of the gene. Remember, 100% or 1 equals totally conserved that position. I then went ahead and inserted a column for the base position because Python starts at 0. And that annoys me when I'm looking at genomic stuff because everything starts at 1. And then use Seaborn and Matplotlib, some aesthetics down here. And we are looking at the S gene conservation across the board. Okay, so this kind of just lets you eyeball things. And we have some areas of conservation here. We have a big area here that's worth targeting. We also have a lot of areas where there are mutations, you know, sub 82% amount of conservation. Just something as a point of reference for us when moving forward. So I thought we'd go ahead and do the same thing and look at the more mutated gene, or the NSP3 gene. I'm sorry, less mutated gene. I did the same exact thing using Seaborn and Matplotlib. And although it looks like there are more mutations across the board, there are, because this is a larger gene. Remember, this one was around like 6,000 bases, whereas the S gene was only about half that size. So just eyeballing it, it looks like there are more mutations. And we can see that, you know, like compared to, no, this one should be more mutated. See, even I get confused sometimes. And so we can see that this one, no, it's less mutated. I don't know what I'm talking about. Yeah, see, this is tricky because this is a longer spans of DNA. And so when I look at shorter fragments, I can kind of eyeball it. There's another way that you can look at this, and I show it to you in the course, that uses like colors that's a little more easier to handle for your eyes. But, you know, we have some higher rates of mutations across the gene, which are interesting. Yeah, but nothing really notable at this with this visualization. Like I said, this is really a simple representation of the gene. And for that, that's about it. That's all. I know I moved through that really quickly, and I apologize to you if that was too quick. I promise I do go into more depth in the course. If you do want to take the course, it's called Python for Genomics. I encourage you to check out the data science pass as well if you have some trouble understanding data science concepts. That'll really get you situated in there. And so with that, I thank Remoter for inviting me. I thank you all for your attention. And if there are any questions, of course, I can field them at this time.
Thank you so much, Adriana. It looks like there are no questions on Facebook. We'll give you guys one more minute if you have any questions to go ahead and put them in that Q&A section. But once again, thank you guys so much for attending. You'll receive a copy of today's webinar later on today or first thing tomorrow. You can also check the Data Science Week page, which has that, and just go to INE.com. It's right at the top. Data Science Week, and it will have it as soon as it's ready as well. But once again, thank you guys so much for attending, and keep an eye out for all the rest of our Data Science Week events.